everybody. We started the auditory system, auditory perception, and in last class I already talked about the properties of the auditory stimulus, which is the sound, and we talked about the sound waves, the properties of sound waves, and also we talked about the anatomy of the ear. We talked about the parts of the ear and the functions of different structures inside the ear and how the sound waves are transmitted to the cochlea to activate the hair cells. And you also know there are two types of hair cells, inner hair cells and outer hair cells. Inner hair cells are more important for the sound transmission, processing the sound, because about 90% of the sound, the fibers that carry the sound, go to the brain through the inner hair cells. Only about 10% through the outer hair cells. However, outer hair cells play some important roles to modify or modulate the sound. So sound modulation is an important function of the outer hair cells, because the signal from the brain can come down all the way, top down signal to modify the sound input towards the brain, and it can come all the way down to the hair cells. So here in this slide you see the basilar membrane. You know that inside the cochlea you have the basilar membrane, and the hair cells are sitting on the basilar membrane, and basilar membrane has very systematic organization of the hair cells. That means the hair cells, those are sitting on the basilar membrane, are systematically organized. The hair cells, those are located close to the base of the cochlea. So here is the base, and this is the apex, the free end or tip part, and the hair cells located near the base are the cells, those are tuned to high frequency sound. The part close to the base is more responsive to the high frequency sound, and as you go towards the apex, the frequency gradually decreases, okay, systematically. And if you look at the hair cells or part of the basilar membrane near the apex or free end or tip, the hair cells in this part are sensitive to low frequency sound, so high to low. So we can say that different parts of the basilar membrane are tuned to different frequencies of sound. Here you see here they have shown the same thing. This part, this is high frequency because the wavelength is short here, right? But as you go this way, you see the wavelength increases. Longer wavelength indicates low frequency, right? Shorter wavelength indicates high frequency. For example, this one has shorter wavelength compared to this one. So this is wavelength is longer. So high, this is low frequency, this is high frequency, and that is actually indicated by the resonance theory, which is also known as place theory or space theory. Same thing. Different frequencies of sound affect different portions of the basilar membrane, which in turn affect different populations of hair cells that I just explained. This is something interesting cartoon. You see that the doctor is asking his patient that the ringing in your ear, I think I can help. The ringing in the ear is called tinnitus. I believe all of you have heard this term, clinical term or clinical condition, tinnitus, which is ringing in the ear, inside the ear. So the patient often hears a sound, although there is no source of sound around. So something, triggers that sound without any sound stimulus from outside. How that can happen, exact mechanism or reason is still unknown. However, we can speculate that intrinsic activation of the hair cells, as I mentioned before already many times, that your brain is sitting inside a dark cranial cavity. It doesn't know what is happening outside. It only knows how to count digitally the signal, how many times the signal is going to which area, functional area of the brain. Okay, so your auditory cortex counts how many times it is getting the electrical signal from the hair cells. How the hair cells are being activated, brain doesn't know. No way to know it by the brain. So if without any sound the hair cells are activated intrinsically inside the cochlea, then the excited hair cells will send signal to the brain, to the auditory cortex, and the brain will count it and will give you the perception of sound. So that is how we can explain tinnitus. Some people, it is really very disturbing and terrible if during the sleep, and it happens very often. Sometimes it happens like once in every few months or once a year or so, which is okay, but if it happens very frequently, that is a problem. So here you see the inner and outer hair cells. So outer hair cells are here and inner hair cells are here. Okay, and these are the ganglion cells, spiral ganglion cells, and ganglion cells are located in the ganglia. You remember in last class I mentioned that mostly bipolar neurons, sometimes pseudounipolar, they receive the signal from the hair cells, both inner and outer hair cells. However, one thing you notice here, that more ganglion cells receive signal from the inner hair cells, okay, than the outer hair cells. Only few ganglion cells, spiral ganglion cells, receive signal from the outer hair cells. Okay, and so here these are the ganglion cells receiving signal from the inner hair cells. Okay, and this one is from the outer hair cells. So if you compare, more ganglion cells are receiving signal from the inner hair cells. Okay, and one ganglion cell receives signal from multiple outer hair cells. Sense? And another thing you see, that from the ganglion cells, of course, the signal will go towards the cochlear nucleus, and the bundle of the axons of these ganglion cells form the cochlear nerve. Okay, so the axons are the cochlear nerve fibers, which is a part of cranial nerve number 8, auditory nerve, and so these fibers form the cochlear nerve and take the signal towards the brain. However, this is something you see here. From the brain, top-down signal, as I mentioned a few minutes ago, also come down and go all the way to the outer hair cells. You see here, many outer hair cells are receiving top-down signal from the brain, and also the afferent fibers. These are the afferent fibers bringing signal towards the ganglion cell. you see the top-down signal also goes to the afferent fibers of the inner hair cells. Why the top-down signal is coming from the brain all the way down to the hair cells? The function of this top-down signal is to modify the afferent signal that is going towards the brain. So if the brain thinks that the signal is arriving, needs to be modified, it can send signal downwards to different relay stations as well as all the way down to the hair cells. Human cochlea has about 33,000 spiral ganglion cells, and about 90% of ganglion cells innervate the inner hair cells that I mentioned, and about 10% innervate the outer hair cells. Okay, cochlear branch of auditory nerve is formed by the axons of the ganglion cells, and mainly by the axons from the inner hair cells that I mentioned already. And now the fibers that take the signal to the cochlear nucleus, the cochlear nerve fibers. The fibers could be three different types. Lieberman, a scientist in 1978, he presented evidence that auditory nerve fibers could be classified into three groups on the basis of their spontaneous firing rates. Some fibers have high spontaneous firing rate, that means without any stimulation, spontaneous, and medium spontaneous rate fibers and low spontaneous rate fibers. High spontaneous fibers would fire between 18 and 250 spikes per second, and most of the fibers are high spontaneous fibers, okay, 61%, and medium spontaneous would fire 0.5 to 18 spikes per second. So it could be like, 1 spike 2 seconds, that's the lowest, and 18 spikes per second. 23% fibers are medium spontaneous rate, and low spontaneous would fire less than 1 spikes per 2 seconds, okay, and about 16% fibers are the low spontaneous firing fibers, okay. And the firing rate or the number of action potential would depend on the size of the synapses, okay, size of the synapses on the inner hair cells. If the synapse is small, the resistance is more, and if the resistance is more, the firing rate will be less, okay. In larger synapses, the firing is more. These two scientists are very well known in especially in the field of auditory perception. One is George von Baxi, and he won a Nobel Prize, and he actually proposed the space theory, resonance theory, as I explained before, that different parts of the basilar membrane, right, is stimulated by different frequencies of sound from high to low. The area near the base responds to high frequency, and near the apex low frequency. So by looking at which area or part of the basilar membrane is responding or moving, we can tell that what kind of sound frequency is that. Make sense? So if you see that this early part, base part, is moving, that means the high frequency hair cells are being activated near the base. If you see the apex part is moving more, that means the sound presented is a low frequency sound and activating the low frequency hair cells. Okay, so by looking at the part of the basilar membrane which is moving or creating waves, by looking at that we can say which hair cells are being activated and what type of stimulus, sound stimulus is that. Is it high frequency or low frequency or medium frequency? Makes sense. So that is studied by George Bexey, and he used the silver flake, and he also used the strobe photography as marker. So by observing the marker, he was able to tell which part of the basilar membrane moved, okay, like surface wave, and that's how he studied the activity of the basilar membrane and proposed the resonance theory or space or place theory. Okay, he was awarded the Nobel Prize in Physiology in 1961. Another famous scientist in this area is Ernest Glenn Weber. And Glenn Weber, he suggested another very important theory that is called the Vole Theory. And what the Vole Theory says, the Vole Theory actually explains how high frequency sound is transmitted through the cochlear nerve fibers towards the brain. He actually suggested that it is not necessary for a single fiber to fire an action potential every cycle of a sinusoidal sound signal. That means multiple neurons participate together to carry a high frequency signal towards the brain, because a single neuron or a single fiber cannot carry alone a very high frequency sound, which is understandable, and that's why he said that a population of several fibers, neurons, behave independently, work independently, and the frequency can be coded by volley of spikes carried by those multiple neurons, that each fiber on only some cycles, only few spikes per second. Okay, so let me explain the Vole theory. So this is your auditory cortex, and you know that you have frequency map, and this is very high frequency, this is low frequency. Now this is the basilar membrane in the cochlea, so high frequency here, low frequency here near the apex, right? You already know that. Now we know that the range of the sound we can listen is 20 Hz to 20 kilohertz. That means 20,000 Hz. It's a very high, right, number per second. So 20 Hz, a single nerve fiber can carry this low like 20 times per second, which is possible. However, how about like 20,000 spikes or action potentials per second? Can a single fiber can take it or carry it? It is impossible for a single fiber to carry that high frequency. Okay, therefore multiple fibers work together, population of fibers work together to carry that high frequency sound towards the brain. Now how they work together that was explained by the Vole principle or Vole theory by Dr. Weber. What he explained is that one fiber, for example, this one is firing at this rate, okay, fixed interval, of course, and this is fiber number 2, and this one is firing like this at different times. You see here I am showing the interval is same but at different times, right? And another fiber number 3 is firing at same interval but also slightly different times, little bit off from each other, right? So you see here, this is spike number 1, this is spike number 1. So spike number 1 in fiber 2 is firing slightly later than spike number firing of fiber 1. Same in spike 2, you see here, and this is spike 2, right? After the fiber 1, not immediately, not at the same time. Spike 3 also at slightly different timing. Now, when I add all these 3 fibers together, I'll get that the action potentials or spikes are being carried towards the brain by those 3 fibers together is the combination or summation of those 3 fibers, right? So high frequency. So that's how I just gave you the example how 3 fibers work together combinedly to carry the signal, which is a high frequency signal, towards the brain. Okay, so if I add these 3, I'll get high frequency signal is being carried towards the brain by those multiple fibers. So that is how the Vole principle explains brain rules for auditory analysis. Now we already know that how your ear processes the sound waves and how the hair cells are activated and send signal to your auditory cortex to give you the loudness and or pitch of the sound, right? We know that each ear is enough to process that auditory signal. So even if you close your one ear, with one ear you can understand what is you are listening, what another person is talking. However, why you have 2 ears, not one? We have 2 ears to give you more information about the auditory scene. That means what are the things going on around you, not only understanding what somebody is saying or what kind of sound you are listening, what is the frequency, is it high frequency, high pitch, or high intensity or high loud volume of sound. That can be processed by the signal going to the brain from one ear. However, you need 2 ears to do further auditory scene analysis. And auditory scene analysis includes the sound localization, okay, which is the most important part of auditory scene analysis, and you need uh, location of the sound or localization of the sound. To do that, you need two ears, and your auditory system has some limitations because it is not as precise as your visual system. Using your eyes, you can specifically locate, right, any object in terms of angle, okay, and very small difference of the location of the object or size of the object, you can easily discriminate, even they are very close to each other. However, your auditory system is less reliable or less precise to do that. However, remember that your visual system has limitations too. For example, we are frontal-eyed creatures. That means our eyes are located in the front of the head, and we have limited visual field, both horizontally as well as vertically. Our field is limited. We cannot see what is going on outside of our visual field unless we move or turn our head sideways or up or downward. So that is a limitation. However, in case of auditory, you don't have any space limitation. That means sound can arrive into your ears from any direction, right? Also from the back of your head, from the top of your head, above the head, below the head. So the field is like 360-degree wide field. That is an advantage of auditory. If someone comes or approaches towards you from the back to harm or to do something, you can listen that sound and tell that. But if you don't listen, your visual system cannot detect that. So both have limitations and advantages as well as disadvantages. But one thing which is interesting, that auditory system processes more complex form of signals. That means the sound, as I mentioned before, we listen is a very complex form of sound. All the sounds you hear are complex sounds. Of course, in a party where many people are talking to each other in different groups, some are eating, some are talking, some people are opening the door or closing the door, all those things, you cannot stop listening those things. In case of your visual, you can close the eyes, right, and you will not see anything, so you can avoid everything. However, in case of auditory, you listen everything, all those signals, auditory signals are entering into your ears simultaneously in a very complex form. That means your auditory system is accepting or receiving the signal in a very complex form. That means when the signal is being processed in the brain or central nervous system, this part of the brain responsible for auditory processing is performing more complex function to process that complex signal. Makes sense, right? So auditory scene analysis is a very complex mechanism or process in your brain. And the term selective attention is one of many properties of auditory scene analysis. This is just, remember, this is just one. Selective attention, we can selectively pay our attention to listen a particular thing by ignoring other things going on. Okay, as you know, in a party where many people are talking to each other, in a small groups, and if suddenly you listen that somebody mentioned your name, in that crowd, you can selectively pay attention and ignore other stuffs are going on around you to listen what those people are talking about you, or what that person is talking or mentioning about you. We do that, right? So that can be done to do the auditory scene analysis, what is going on around you in the auditory scene. So that is one, but there are other ways to analyze the auditory scene. We'll talk about those. Another one is stream segregation, which is also very STREAM, stream analysis or segregation, auditory segregation. We'll talk about those. And this is called the cocktail party effect. Here you see the ascending pathway of the auditory signal. So how from the cochlea hair cells signal is taken to the primary auditory cortex, all the way to the primary auditory cortex. You will see that at different locations or different stations, the signal is processed, and in some areas the signal is taken to the contralateral or opposite sides. That means the crossover take place. Signal is taken from the same side to the opposite side, and that's how both sides of your brain, or both hemispheres, both primary auditory cortices, are receiving signals from both ears, which is very important. As I mentioned, that your two ears are important for auditory scene analysis, including sound localization. So you see here, first the auditory nerve, which is vestibulocochlear nerves, and the cochlear nerve fibers, axons of ganglion cells, right, take the signal to the cochlear nucleus here in the medulla oblongata in the brain stem. Okay, so cochlear nucleus. So this is the cochlear nucleus. Okay, and then from the cochlear nucleus, the signal, the processed signal, is taken to the midbrain. And which part of the midbrain gets the signal? That's the superior olivary nucleus, or superior olive. So this is midbrain, this is medulla, cochlear nucleus here, superior olivary nucleus or superior olive. Okay, here you see that many fibers are going to the opposite side, opposite olivary nucleus. That means crossover take place. So from one side to another. So we can say that the signal from two ears are combined in very early part of the auditory pathway. Then this is something different than the visual system. In case of visual system, the signals are taken separately, and they start to join together in very upper part, in the upper part of the nervous system. However, in case of the auditory signal, the right and left ears signals are combined at the very early stage of the pathway, or early part of the auditory pathway. Okay, then the signal goes through the lateral lemniscus. So lateral lemniscus, here some relay take place, and then from lateral lemniscus the signal is taken to the inferior colliculus. This writing is not very clear. This is inferior colliculus. Okay, and it is also in the midbrain, and in this area you also see some crossover. That means signal is being taken to the contralateral side. Okay, and then from the inferior nucleus the signal is taken to the thalamus, and in the thalamus, as you know, thalamus is the major sensory relay station. That means, most of the sensory signals are processed in the thalamus before it is taken to the respective cortices. So the part of the thalamus that processes the auditory signal is called the medial geniculate body, or medial geniculate nucleus. Okay, body or nucleus. And then from here the signal is taken to the primary auditory cortex, you already know, located in the upper part of the temporal lobe. So that's how the signal is taken all the way to the primary auditory cortex. Okay, so here just a little more explanation about the cochlear nucleus. So from the cochlea, the signal, cochlear hair cells, the signal is taken to the cochlear nucleus first, which is in the medulla oblongata, and signal, auditory signal carries the temporal and frequency information, of course, and those are processed in the cochlear nucleus. So you see that they are being processed first in the cochlear nucleus of the medulla oblongata. Okay, and then in the superior olivary nucleus, or olive, as I mentioned, that significant crossover take place. Okay, the neurons of superior olive are getting signal. Since the crossover takes place, the neurons of both sides, superior olive, get the signal from both ears. Makes sense? And that's why this area, the superior olive nucleus or olivary nucleus, neurons are involved in sound localization, because the signal from two ears should be combined to help you to localize the sound, right? Two ears are necessary. Then the axons will pass through the lateral lemniscus, right? Then inferior colliculus in the midbrain. Okay, so some crossover also takes place here, as I mentioned. It says major crossover take place here in the inferior colliculus. Okay, and then to the thalamus, where the sound signal will be processed in the medial geniculate nucleus. Okay, and Thalamus, always I mentioned at the very beginning of this course that thalamocortical, right? This is thalamus, this is the cortex, and thalamocorticothalamic feedback is very important. So from the thalamus, signal goes to the brain, cortex, not brain, to the cortex, and from the cortex again feedback, uh, significant feedback returns into the thalamus. Okay, signal returns into the thalamus, and that is thalamocorticothalamic. Okay, this thalamocorticothalamic feedback is very important to keep us awake, to keep our brain function active, our consciousness. Okay, um, so that is very important. And then the primary auditory cortex. Okay, so now in the cochlea, in the cochlear nucleus, where the signal is first processed, as I mentioned, that both frequency and temporal codings take place there. That means you have two types of neurons, stellate neurons or cells and bushy cells or neurons. And the stellate cells of the cochlear nucleus are like tonic firing, and the bushy cells like produce bushy cells produce the phasic. That means two types of firings. We talked about these before. That tonic type of firing, which is the property of the stellate cells, would help you to determine how long the stimulus was present, because as long as the stimulus is there, the stellate neuron will continue to fire. So by looking at this firing, we can tell that how long the auditory stimulus was present in terms of timing, how long the signal was present. But another very important thing, this stellate neuron is telling you every time the auditory stimulus, one stimulus or tone is presented in that auditory stimulus, the neuron will fire. So that means the frequency will be also given by this firing, because if this is 1 second, so that means this is 5 hertz sound, right? 5 times. So stimulus in this auditory stimulus, you have 5, the frequency is 5 hertz. That means 5 waves of stimulus. So 5 times the cell is firing. So how many times the cell is firing? By looking at that, we can tell what is the frequency of that sound. If that sound stimulus has 20 waves, then this neuron will also fire more. That means will fire 20 times. So each wave would stimulate, will create one spike. Makes sense? So this is frequency coding. That means how many times the neuron is firing. That tells you what is the frequency of that sound stimulus. Bushy cell cannot give that information, because it fires in phasic way. That means only when the stimulus arrives it will fire. Then even if you continue presenting the stimulus, it will not fire. So only when the stimulus arrives, so that will give you the arrival time or onset. So this one is giving you the time of onset of the stimulus, arrival of the stimulus, and this one is giving you the frequency of sound. Okay, and both are needed. Both are these are two very important properties of the sound, right? And therefore you have two types of cells there to process them separately. Okay, now auditory scene analysis is a complex task, as I mentioned, highly complex task, because the sound we listen is a highly complex sound. I gave you the example. In a noisy place it is more complex. However, even if you are sitting in a quiet place like inside the library, where you may think is very quiet, but just stop reading and just try to concentrate your mind and try to listen if there are other sounds you are listening. Now you will be able to realize that not this place is not really absolutely quiet, because you will be able to listen the sound of the heater if the heating system is on, air conditioner, right? Or like if someone is moving the chair, okay, or opening the book or closing the book or putting the book back in the shelves. All those weak sounds are always present around in that area. Although when you concentrate, as I mentioned, that selective attention, when you are focusing on your stuff, reading or writing, your auditory system has the ability to filter the other sounds. So that's what happens, but the sound is also not simple. It is a complex sound you are listening, even in a quiet place. Okay, so sound localization is a very important aspect of auditory scene analysis, and the cues of the sound that we listen help us to localize the sound, includes the ITD, which means inter-oral time difference. Inter means in between, oral, auricle, ear, time difference. That means the right and left ears. If this is your left ear, this is your right ear, okay, and this is the source of sound. So sound arrives into your right and left ears, and the difference of that arrival of sound is the interoral time difference. Okay, interoral level or intensity, same thing, difference, ILD or IID. Okay, interoral level of difference or interoral intensity difference. That means the pressure difference. That is another important clue or cue to localize the sound. Okay, the onset, the arrival time of the sound is very helpful to understand what is going on around you, what are the things going on in the scene, auditory scene. The frequency, of course, the change of frequency or the change of the pattern of frequency, the intensity, the change of pressure, and the timbre. Even the frequency and intensity are same, the sound you would listen could be different. That means the quality of the sound could be different, even the frequency and intensity are the same. So that is called the timbre. So all these are being used, or all these are used by your auditory system to give you the perception of auditory scene or auditory image. What are the things happening around you? Okay, let's talk about the three dimensions of sound. When we localize the source of sound, your auditory system measures three things, or three measurements. One is the location of sound source in x-axis, that means horizontal position, if it is right, left, or just in front of you. Okay, and how much right or left, and that is the horizontal plane, you know that, and that is called azimuth. And another is elevation, up down, how above or below the sound is. Okay, that is another dimension, that is called the elevation, the vertical position, okay, or movement of the sound source. And another is the z-axis, that is the distance of sound, is it near or far. Okay, so that direction is the distance. So all those three are combined together to help you to localize the sound, from where the sound is coming. Okay, and this is also distance, how far or near, is also called the range. Okay, range. So now if the sound, this is your head, okay, and if the sound is just in the center, okay, that means your right and left ears will receive the sound at the same time, because same distance here. Makes sense? The distance is same, so the arrival time will be exactly the same through the ear, okay, into your right and left ear. So the interoral time difference will be 0. Make sense? Now if I move towards left, the sound source from here to here, then you see your left ear will receive the sound waves before the right ear. So the difference will be more than 0. Make sense? The arrival time, if this is 3 seconds, this is 2 seconds, what is the difference? 1 second, right? But in this case it is in the center, that means no difference, 0. So as you move towards, sideway more and more horizontally, the difference will increase. in both ways same. The difference will increase. Okay, and also if it is just in the middle in your back of the head, same difference, 0 is here, right? But as you move that way will increase because it is getting closer to one ear but farther from the other ear. Okay, so that is a very powerful cue to localize the sound in horizontal plane in azimuth interaural time difference. Okay. That is what you see here in this picture, that this is 0 microsecond difference just in the center. That means 0 degree, and the interaural time difference is 0. And as you move laterally more, you see the difference increases. Here towards left minus 200 microsecond, same distance towards right 200 microsecond. As you move more, the difference increases, right? And at 90 degree, that means straight right or left to your ear, the difference is highest. Makes sense? So if the difference is highest, that means it is just straight lateral to your one side of your head. Again, in the back, straight in the center is 0. The difference is 0. Okay. Now you can just ask me how you will know, your brain will know, because if it is just in the front, the midline, in the center, it is 0. Also just in the back, in the midline, in the center is 0. How your brain will know, because brain cannot see what is going on outside, right? Sitting in the dark cavity. How the brain will know if it is straight in the front or in the back? That is helped by another cue. When the sound waves will pass through the air, when it comes from the front, it will hit your face first, right? In your face you have multiple structures, your nose, your cheek, chin, all those, and then will enter into the ear, same this way, right? But when the sound will come from the back, it will not hit the face, it will hit the back of the head. The structure is different there, and then will enter into the ear. So we learn that your brain learns to determine that difference of the pattern of sound that is entering into the brain. Okay, and by using those clues or cues, your brain can tell if it is coming from the front or from the back. Same thing if it is coming from above or below. When the sound is traveling into your ear from above, it will hit your head, and then will hit the upper part of the pinna and will enter into the ear. When it comes from below, it will hit your chest, your body, shoulder, your face, and then will enter into the ear. And that is called the head related transfer function, HRTF. Okay, another important cue for sound localization. Okay, so some of these cues your brain learns from experience, right? When your brain develops after you born, uh, your brain can segregate or separate those and can interpret them separately to localize the sound. In the auditory pathway, you have the neurons. Those are specifically tuned to interaural time difference and interaural intensity or pressure or level difference. And because of those neurons, your brain can detect the difference of arrival time, right? And that helps the brain to determine the space or location of the sound source. So we say those neurons are combination sensitive neurons. Sensitive neurons, right? So they are excited by particular time difference. Like you have a map here, we'll talk about that soon. You have a map here. These are the neurons in your superior olive, okay, olivary nucleus. You remember where the signal from right and left ears are combined, okay? And this is a kind of space map. And if this neuron fires, then probably the difference is 1 microsecond. If this one 10 microsecond, this one 30 microsecond. So different neurons fire at different interaural time or intensity differences. So which neuron is being fired? By looking at that, your brain can tell the location of the sound in space, because you know uh, uh, at different locations the interaural time difference or ITD will be different. Also the pressure, amount of pressure will be different, right? If it is close to the left ear, the pressure of sound will be high in left ear than the right ear. Makes sense? As it travels far. Frequency will not change. Two things will change. The pressure will be more in this ear because it is near, the sound is near. Pressure will be less in this ear because the sound is far. But the frequency will not change. Another thing will change is the time, right? So those two things will change, time and the pressure or intensity. Okay. Distance plays also an important role to localize the sound or to determine the auditory scene. The good example is here. You see, a person is playing, standing on a stage, and audience, and audience sit at different distances, and the sound, if the audience is here, a person is sitting here, he will get, you see, more direct sound from the player or performer, okay, less indirect sound. This is direct, okay. Now if you see this person will get less direct sound but more reflected sound. Okay, that is more in case of this person or this person, because the sound is going to all different directions, right? And the sound waves are hitting many structures before they are arriving into the ear of this person, hitting the other people's, hitting the walls, hitting the roof, ceiling, and then entering into the ear. So you see, the sound waves arriving into the ears of this person is definitely different than this person. This person is getting more direct sound from the speaker or the performer. This person is getting more indirect sounds. Okay, and that is one important cue, that how far the player or the performer is, or the speaker is, how many direct and indirect or reflected sounds you are listening. Okay, so if more indirect or reflected, that means distance is more. If more direct, that means the distance is less. Another example is the change of intensity over the time. Like, when you sit in a stadium to watch the car race, you know that cars come towards you, the sound increases, then just passes you and go away from you very fast, the sound goes down, right? Intensity, pressure goes down. Again, the cars come towards you, the sound intensity increases, and then goes against you, the sound gets intensity gets weaker. So this is you, the car is coming towards you, then passing you, and then going away, and again coming, right? So you see that intensity increasing, intensity decreasing, right? And by comparing the change of intensity, your auditory system is determining that if the object is coming towards you or going away from you, right? Based on the timing over the time. So that is another very important cue, right? The change of intensity or pressure of sound. Another good example could be like, you at night, if you hear the sound of frog is calling, and if you listen that the sound is getting more loud, that means the frog is hopping towards you. Okay, if you hear that the sound is getting fade, that means getting away from you. Without looking, your auditory system can detect. So all these cues are helping you to determine the location of the sound, and these are helping you to determine the distance or range of the sound source. Okay, so let's stop here, and we'll talk more about the auditory scene analysis, especially in the complex auditory scene analysis in next lecture. Thank you.